Hello, I'm Will Howat. I'm the head of imaging and immunohistochemistry at Abcam. I've been at Abcam for the last 7 months, but my career to date has been, for the last 20 plus years, I've been in academic research. At least 11 of those were in cancer research biology. And I've moved into industry in the last 5 years. So as a brief overview of immunohistochemistry, it's using antibodies to discover proteins, whether in a cellular context on sections of tissue on a glass slide. So we use antibodies along with secondary conjugates and polymers in order to be able to visualize those proteins, either through a bright field, a normal microscope, or in a fluorescence setting using a fluorescence microscope such as a confocal or other fluorescence scanning system. And that's something that we commonly do every day and is run throughout the UK and the world. It's the same way across the whole world in terms of the methodology that we use, and that's been run in clinical hospitals and clinical laboratories as well as in research labs everywhere. So the current challenges in IHC are around the quality of the validation. So what we need to ensure is that when our researchers are using antibodies, that they are as well validated as they can be. So the research papers that are being produced in Nature and Science and all these fantastic journals are of the highest possible quality. And for that, we spend a lot of time and effort in ensuring that we put a lot of detail into the upfront validation of those antibodies before they are launched onto the Abcam website. So on that basis, we will do a lot of validation. So we will take those antibodies and we will test them in a number of different species, both in human and in mouse and rat. We will also test them across a number of applications, and our key applications are Western blot, immunohistochemistry, immunofluorescence, and my love, which is immunohistochemistry. And so when we're in the immunohistochemistry field, it's important for our researchers to understand that we test these on tissue microarrays that consist of at least 36 different cores of tissue. And there'll be multi-human tissue microarrays of a number of different key tissues, as well as a multi-tumor tissue microarray, which are the key indications for cancer research, be those breast cancer or bladder cancer. And we always test throughout the process of developing an antibody, we will test those antibodies across all of those panels of tissues so that we can be absolutely confident that when that product becomes launched onto the Abcam website, that we know it stains, we know how it binds, and we know it is valid for the target that we've made it for. So what I love about IHC, it's a fantastic visual system to be able to look at what's actively going on in a whole body system. So if you take an individual tissue type, there are a number of different cell types that are present in that organ. And you're able to look at the distinct proteins that are being expressed by distinct cell types and how they interact, those proteins interact with each other, but also how those individual cells will interact with each other. And in a cancer biology field, that's becoming more and more important, particularly in immuno-oncology, where you're looking at the particular presence of certain cell types next to or within the tumor, at the tumor boundary. PD-L1 is a great example of one of those checkpoint inhibitors that's currently in vogue for cancer research. And being able to see that data and visualize it down a microscope with some, whether it's in fluorescence or in a bright field mode, is fantastic because it jumps out of you. When you get a good quality antibody, you can clearly see how well it stains against any background with no non-specific binding, and it drags you into looking at that further. So in the past, immunohistochemistry was based around a single-plex methodology. And that would normally be using diaminobenzidine, DAB as we call it, to produce a brown colour along with a hematoxylin to stain nuclei. But that's now evolving further so that there's a number of different companies that are producing some very high quality either kits or products where you can plug and play your own reagents into to be able to do further multiplexing. Now that can be to 4-plex or beyond. So some of the systems that are there are going beyond that to 40-plex or 90-plex. And so then we're in a system where for a single slide you can look at anywhere up to 40 to 90 different combinations of antibodies and look at the interaction of those antibodies on a cell, as well as I said earlier, on different cells and how those different cells interact with each other in themselves. So we get into very huge amount of data that then needs to be deconvolved into its individual elements so we can make an understanding of what that is, whether that's in immuno-oncology or neuroscience or any of those other areas. We need to spend more time getting that data together at conferences such as AACR and to be able to work out what that means for cancer research. And I see that the future will be that we will go beyond a single color IHC, and in the future within clinical departments and hospitals they will be using a multiplex IHC technology, whatever that may look like. So the implications for multiplex IHC within the kind of bench-to-bench side model is that we are able to then look at more markers. So within that markers, whether that's 4 or 40, we're able to have a more informed decision on the patient stratification. So where we know that patients are heterogeneous in a population, we know that they will behave differently to the drug combinations they're going to get. If we can use multiplex IHC to stratify patients more effectively, that therefore means that we can treat them more effectively in the future. And hopefully that minimizes their side effects and they get increased patient benefit.
My name is Brendan Collins. I've been at Abcam about 6 years. Before that, I got my master's degree in neurobiology, studying circadian rhythms. The customers that I work with are facing challenges of finding a good biomarker signature in a way to not have an invasive technique like a biopsy. So if you're able to get a signature from plasma or serum, that's a non-invasive way to get a patient profile of a possible disease or even a disease before it happens. I am the field scientific specialist that supports the Fireplex product line at Abcam. And this is a product line that you can multiplex up to 65 different biomarkers in a single sample from a very small amount of sample, which is really useful because sometimes with mouse and rat, you don't have a lot of CSF sample, for example, or saliva or plasma serum. So from that little amount of liquid biopsies, you're able to get a lot of data. And from that, scientists can come up with a biomarker signature that can, find a disease ahead of time. We've had customers say that they can find a signature of microRNAs, for example, that might be 2 years out of getting ovarian cancer. And if that's like a game changer in the field. So I support them by, we provide the reagents that they need. We don't require any dedicated instrumentation. We take the instruments that they already have in their lab, and we provide quality reagents to them that will work on those instruments. Well, customers need the reagent. They need to trust the data, and they need reagents that are reproducible. They need to have reagents detect low levels of proteins or microRNAs in a sample from a low sample amount. They need to trust that when they buy more of that reagents later, that they have no lot-to-lot variability. So with our reagents, they are able to profile microRNAs, profile proteins in all sorts of liquid biopsies, but even in tissues and cell lysates and cell culture supernatants. I find it exciting that researchers are able to discover these signatures that are going to detect diseases ahead of time and that could possibly save lives. It's only been around for a couple years now at Abcam, and we've made more and more panels, starting with cytokine assays, but now we're moving into other fields of research to support more customers. Well, I think we were trying to evolve the platform to work on more instruments, broader, larger panels, and even larger number of samples at a time. We are going to provide the tools for customers to get a lot of data, and from there they can drive precision medicine forward.